Another is kanina akong record. Ako pang iwan. Ito ni, atong ikuan niya. Okay. Naraon sa documents. Butang mo na yung date.
Para siya mga part. Ito sa kalim. Sa isa po.
So wala pa man ang uban, no? Or di na to sila mamalik? So supposedly this afternoon,
Hi, it's Mr. Anderson, and in this podcast, I'm going to talk about plant nutrition and transport of this kind of circle. And so Basin has, I don't know if you've seen the Lord connected through these hydrogen bond here. So it's going to move into these.
So by the way, since gawas na lang man siya sa ganina ra ko pinangitaan ni kasi so since gawas na lang man siya dere sa ko ang ano so ito yung mga few pod na mga topics na gusto na ako i-review, no? As we have discussed last time. Yung kanila. Ilagin ako okay, yung mga scientific names nila, sir na, pugoy copy na ihatag na nasa GC. And hopefully ma-print na to karon. Aha naman po isa. Hindi ko kasabot kung ano. So, nasendan ako na sa inyong copy. So, that's my own compilation of the different scientific names, family names, and also kung anong region ang top producer in the Philippines. Hello, Gab. Gab. Kanandali siya kadali na rin. Thank you. Kani kaya akong isend sa Ali. I-print niyo ni Daan, i-backlit niyo po sa file sa ng diretsyon. Kani-ganing.
Nasa na start?
Kung private. Private, hindi. Kaning untitled na po. I-print mo yung una ka ron. Makakat sa ilahat sa sila. Bisenta na ako. Pero ang buhat mo sa akin pagtanawan ko niya. Pag-open nila mo sa inyo ha. Teka to siya, di na siya kalbalo. Kaya passwords. Hindi na yung ma-open sa ato. Ang laptop nila. Then katong uban pag-isend na ako. Then naka-word na siya. I-print niyo after nasa na. Para madala na nila. Ang kanang akong isend sa inyo. Okay, so gamay lang na review about scientific names and family names and top producers. So 40 lang man na siya, and hopefully this 40 dili nato ni siya ma-miss in case na ma-mention siya sa questions in the board exam. Okay, let's say for example, when we say coconut, what's the scientific name? Family. And of course, top producer, Ana. Okay, when we speak of coconut, so under po sa palme or ari kasi, magka-family lang po sila sa ari kasi. Sarata sila. Okay, of course, sa rice and corn, they are both name. And Sharon, makalimtan po ninyong Oriza, Zayamis. Pero pag rice, kinsa ang ka-producer? Wait lang, kay marag wala pa ko na. Sorry pala sa mga online, nakalimot na po ko o share. Sorry. Naka-record na siya. Good afternoon once again and sorry sa mga naka-online kay nakalimot ko share. So, nag-review lang me for on this topic on makita na ba ninyo?
Yes, ma'am.
Thank you, Sir Ron. So again, pag-rice, Central Luzon, yun na siya, no? And how about corn? And for
Sorry.
banana naman,
Davao region.
say, unsa man gyud para di siya ka, balbisiana. Pag Cavendish, unsa siya? Busa para di siya ka. Pag ano, Cavendish, unsa yung isa? Cardava siya, unsa siya? Musa,
Sa Pintong.
Balbisiana. And of course, top producer is but for
Davao region.
calamansi, scientific name for calamansi? Yeah, even scientific name, no? Pwede siya Citrofortanella mindis and citrus limon. Ang limon kasi, dili na siya, citrus limon is not really calamansi. Lahi na siya, lemon na siya nga variety. And still under, so, unsa ning maropa? Ha? Unsa ang maropa? Misamis? Marindoque? Ay, Mindoro tayo, sorry. Sorry, God. Romblon, Palawan. Wow, top 1 si Sir Marlon. And for durian, family, of course, Davao region. Sa arm naman, Lanzones po ang top producing crop, no? Scientific name, family, and for manga, say top producer, family, scientific name, nabuto the eye. And for mangosteen, scientific name, family, and pareho sa Lanzones, arm. Okay, sorry for that. Parang sarap kumanhi. After po. Okay po. Okay. Actually, naka-include na niya siya sa katong classification na fact sheet na akong gisend sa GC. And nagpa-print na lang po ko para next week na ba ako ang ko. Pero napay ugma. Dapat ko ugma makuha na niyo ng fact sheet para mas tadihan ninyo. Post-test na tayo next Saturday for website. Kadali ra sa panahon, no? Pagayta ka pa sa ansay. Okay, and of course, nahinahinaya naman ito yung basa, no? So as I have said again, once you have your fact sheet na, kanilang nga page, pakibasa lang to like twice or 3 times. So, pag mag-memorize mo, diba, basahin niyo 5 times, makuha na sa inyong brain. Ang alin? Wala. Kasi based sa, Philippine Statistics, ang ginagamit nila is kaning by region talaga. Na siya sa PSA. Na katong, ipakita mo na ako sa inyo last time. Pero may bala naman ninyo, diba? For example, kung say Northern Mindanao, kung pineapple man lang, pineapple yun. Ilocos region. Last year ata. Dili last year. 2016. Sa Baguio? Ha? So ang updated is actually, that's why wala po nag-rely sa updated. Di ba nagpakita ko sa inyo sa update? Kasi ang question po for this board exam, dili po siya updated. Sa kanima, Ha? 26. Dili ba ba updated atong PSA, diba? 2019 kani? Ang baseline. Mga data. Kung kano sa ang last na So last time pakita na ako sa inyo, ha? So double check na to, maghimo lang kong outline, no, para hopefully before ta mag-post test. I'll give you katong latest para ma-compare na to ang difference. Diba? during our presentation na may mga naglahi nag-update. So, i-google lang ninyo PSA graph statistics. And nalagdari ang data. Yeah, 2016 to 2020 na mga data pa ang latest. And katong akong gi-present is malamang unya, and ibalik na to ha. Take note, last 2021, 2020 pa ang last na edition niya. Pero ang data niya is 2015, 2019. So mo niya latest. So when we say peanut, pangitanong na lang peanut, Rebe. Page 'Wala yung peanut, no? Let's say, ano na lang. Banana as is Japan na kalamansi, page 8. Or corn, page 4. Okay, sa corn, the top producer is for 2020 is Cagayan Valley. No? And then dito sa ating photo for corn, no? Wala po ko'y corn. Sargent. O, layo ra kayo, diba? Nagbago gid siya. Wherein ang Sargent is kapila na lang siya. Ka first, second, third, fourth. Kaso pag craft ani nga exam, alam nga na mag last month pa rin yung na exam, no? So, ito ta magbase sa katong previous na so, magkuha na lang ko, mag-craft ko o kaning base na lang siya sa 20. Oh, dili karong 2022 ba itong edition? 2021 na edition na lang. Yes. Okay, so now as we proceed, okay naman itong exercise na to sa hormones. So part of physiology na dapat po nato mahibalaan are the different plant responses and mga adaptations as parts of physiology and actually plant nutrition. But last time we already discussed phloem loading, diba? Okay. So these are how plants grow and respond to our environment. Okay. Like for example, first tropism, or it is the growth towards the stimulus, or such as light or gravity. Lahat tayo, even plants, respond to growth or to this stimulus like the light or gravity. Now, like for example, the gravity of the earth, diba? Kaya tayo nakatayo lahat, ang even yung ating tanim, the roots grow downward and the shoots upward because of gravity. And light also because we always look for the light for energy. So that's what we call tropism, growing towards the stimulus. So there are also types of tropism. So have you heard this one before? Kani mga Gnostic movements. Na-discuss ninyo sa inyong college na topics? Physiology. In this part kasi mga higher physiology. So malamang ma-miss ni sa college, especially kung nagsigirag activities, no? Sa una pa man face to face, gusto pa kayo mga intrams and mga foundation and a lot of extracurricular, no? And when I say Gnostic movements, it's the response to the environmental stimuli, okay, which is independent of the direction of the stimulus. So, parang predetermined response, no? Environment ang stimuli naman in this movement. So, for example, so let's talk about first tropism, which is a response to an environmental stimulus. So, usually, meron tayong tinatawag na movements by growth, which are directional. So, we call phototropism when the growth is towards the source of the light. So, that's from the word phototropism. So, mo nang mubawg ng tanong padulong sa ang sunlight. So, that's phototropism. And it has been again proven by an experiment with this. For example, no, gibutangan og kaninga the side was actually then kung gi-illuminate ang light is in this source. Lagyan makikaro kung paano. Ay, sorry. So when the light source is from here, so it will grow towards, and this side of this shaded side of the colloquial, no, it is where auxin elongation takes place. So we all have also phototropic responses in bending of growing stems towards light sources. With the kind of light, pwede siyang with blue light wavelength. And pag hindi siya blue, straight lang po na siya. It doesn't bend when the blue light is removed, but it will bend towards the blue light. At a certain blue light, I guess, is 600 or 700 nanometer. I-confirm na natin na mamaya. So, auxin is again involved in phototropism. So we also have Thigmotropism, which is the response of plants to touch. Diba? So lahat tayo are Thigmotropism actually kasi we always can feel the touch. But for plants, it has directional growth response to contact with an object. Kaya mo cling ang vine because of Thigmotropism, like the tendrils. So this is an example of Thigmotropism. Mag-grow siya, kung makatouch siya, dito siya muliko-liko. And we also have Gnostic Movement, meaning another response to environmental stimuli. So we call it seismonasty. For example, when you come in contact with mechanical shaking, no, like kung tandugo na nimong makahiya or may musa pudi ka, sensitive plant, so unsay mahitabo mo close din siya, no, so that's seismonasty. Okay, and ang explanation ana, it has a motor organs, mulihok siya, no. It has the pulvinus at this part. So this is the motor organ, the pulvinus. Ito na ko makita ko ang paano eh. Ay, ayan pala. So the pulvinus is the motor organ. Now, and when if there's in contact with movement or touch, that's the time na mo close siya. And we also have the gravitropism, no? Of course, in response to the a directional response to the force of gravity. So, usually during germination, in which auxin plays the role. So once gravity is perceived by the cell, then there will be signal that perceives gravity and induce, transduce, intra and intercellularly. That's why plants will grow and the cell will elongate. And there will be increased concentration in the lower stems and it will bend. Now this is another gravitropism. So if it bends up against the force of gravity, this is called negative gravitropism. But when the upper side of the roots oriented horizontally will grow more rapidly than the lower side, wherein the roots will grow downward at this point, it's a positive gravitropism. So na yung mga tanong, usually roots lang man, no? But there are, it's the roots, no? We're talking of roots here. So if the roots will grow downward, that's positive gravitropism. Pero na yung mga roots na mo grow upward, that's negative gravitropism. So that's a typical example, no? It will move downward, or gravitropism, also called geotropism. So the cells called as statoliths are the responsible parts na maka-sense of gravity. So cytokinins naman, the growth hormone, will affect plant growth in what way? Of course, through cell division, growth of lateral buds, and will cause dormant plant to sprout. Delay also aging of leaves and inhibit elongation and cause cells to grow thicker. So as a result, the cytokinin of the top caproot is 2 times bigger than the other. So yun yung effect ng cytokinins. And gibberellins naman will produce increase in size sa stem and fruit at saka responsible for early growth in many plants. So as you can see, the differences is within this one and this one with GA, and this one with GA and without. How about ethylene? So for ethylene naman, for leaf loss at saka ripening. And it is also used to treat fruit picking or yung preservation before ripening to ripen them when being shipped or handled for shipping. So this will also show the auxin production in the plant. Nga nung mo grow siya or mo bend, di ba? Epiphyte movement towards the growth of the plant, towards the side of the sun pala, and it's because of the auxin cellular division here, or elongation. So this is explained in this diagram. So ang concentration of auxin here will move for growth in this side, kaya padulong siya sa. And for geotropism naman under gravity, so negative tropism if it grows upward. So how does auxin affect growth in presence of light? So these are the explanation. Una, it hits the stem and the concentration of auxin in the shaded part will go high, which will result in concentration stimulating cells in the dark side to elongate. So Ang result niya, mag-bend siya toward the light, so that's epinastic or epinasty. So, ang auxin naman will build on the lower sides of the roots and stems sa gravity, and the roots auxin will inhibit cell growth and elongation, and therefore roots will grow downward. So in terms of height and width naman, auxin, as the stem grows, it will produce lateral buds. So ang lateral buds will become the branches or side branches. And this growth at lateral buds will be inhibited by auxin on the stem tip para mag-lateral bud siya. So remember now, auxin is also responsible sa apical dominance. So the closer a lateral bud to stem tip, the more also it is inhibited. Kaya mag-lateral growth siya. So those are just reasons of the effects of the different hormones to plant response and growth. So we have also the so-called photomorphogenesis, which are one of the plant's response to light. When we say photomorphogenesis, it is non-directional light-mediated changes, and the responsible light is red, phytochrome red, which will trigger from photomorphogenesis, or where stems go from ethulated to unethulated, no? In dark phytochrome to light phytochrome red, far red naman, PFR. So it will just mag-ano lang siya, no? Mag-form lang siya. For example, mag-form na ang roots, mag-form na ang shoots, that's photomorphogenesis. But for photoperiodism, it will regulate seeds, no? Like lettuce and some weeds. And with phytochrome red, it will inhibit germination. That's why sometimes we need to cover our plants when pagtanong nato. Kasi there are phytochrome red which can inhibit germination. But for example, for lettuce and some weeds, the light will enable them to the opposite, germinate with the presence of light. And the rest will not, no? Inhibit pa noon. And once it's converted to phytochrome red, it will induce germination. So the red light will induce germination, while the far red light, this is an example of the effect, no, of the photoperiodism. And when far red light has no germination, and so take note, pag unsa ang last na na-touch, like i-flash mo siya red, then far red, then red, so mag-germinate siya kasi ang red light is for germination. But maski pa nag-red light ka, pero ang ending mo nga treatment is far red, it's for no germination, so walay germination. So those seeds not buried deep in the ground get exposed to red light, and this signals germination. So kung ilubong mo siya maayo, and it's only red light lang naman, no, na dili nakapos. And when in sunlight, nanay wavelengths ba yan, including red light. So regulates when plants flower either in spring or later in summer and fall. Welcome ka dito. So how phytochrome works, this is actually just a diagram to show you na the functions ng phytochrome. So when there is a biological response, anong gagawin ng phytochrome? With the photodestruction. Once na is the stimulus, no? And then phytochrome will act on the different lights. It's either far red light, so that's 730 nanometer, red light 660, and if it is dark, no? So ano gagawin ni phytochrome? It's lahat ng ano, this wavelengths from 660 to 730 will convert phytochrome far red into phytochrome red. And that's the time synthesize ang amino acids. So that's how phytochrome works. So let's proceed with nectinasty. So these are sleep movements. We have the so-called prayer plant wherein the lower lids during the day and raise, they will lower their lids during the day and raises leaves at night. Kasi ang reason pala na is they are actually preserving transpiration. No? Like the shamrock and the legume. So marag mo, sana so sana marag ka ng ano sila ba? Tree bell. So ang tawag doon ay sleep movements or nectinasty. So have you heard, of course, circadian clocks or rhythms? It's the same example when jet lag ka, no? Ikan ka o abroad from other countries like Israel, abot ng Pilipinas, lahi ba ato ang biological time difference, no? So kung gabi, buntag dere, gabi dito, naanan ka dito, pag-abot dere, mag-adjust yung kag mga 1 month ana. So imo pagyapon ginadala-dala nga unsay time na naandan nimo from other country. So, kinsa naka-experience na ana? Sir, asa kagikan, sir? Ha? Sa Israel kagikan? Last, ano sa year? 2020. So, pag abot nimo dere pila ka kabulan? Dito mo nag-a-adjust. Wala ray bulan. Basta na ray tanduay, no?
Ayaw kang gabi.
Sige. What if you, Dre, jud, no? Mahubog na ka, Dre. Tingala mo punta. Ay, siyem, naka-claim sa ko, ano, na price. Kasi nag-top ba siya last time? Tanduay ito akong ipalit. So, unsa na lang kaya, no, na pag walay, kasi kita naanan ta kagabi, maggabi matulog. Maliban na lang uban na rin mga asa mo nagtrabaho, mga vampira. O matulog jud mo adlaw jud maski walay gabi. Pero ang ubang country nga 24 hours ang adlaw, di ba? Walay gabi. Ay, ina din na lang pud matulog. No. Di ba? Di ba like sa Iceland, di ba? Na ay 6 months sila nga walay gabi. So circadian clocks are the endogenous timekeepers, not only for humans, animals, but also for plants. So it's the plant will keep plant response synchronized. Parang naandan na nimo ba, no? Walang ang attitude nato pag naandan na, lisod na gina siya i-change. No, feeling nato ay di na yun na, wala na pag-asa, di na mabago. Pero mabago good na through training. Diba? Sa una, sige mo kalit sa trabaho, sa eskwelahan, hantod na lang jud nagtrabaho. So dapat i-change, no? So ma-change mo na mo matagam, diba? Diba, Dre, wala kayo nag-inom? Diba, wala na kayo ka nag-inom? Panagsa. So, kahit nakatrabaho ka o nakagraduate ka, Dre, perminti na, dili na panagsa. Kada adlaw yung college, grabe. Maghilak na lang ang adlaw. Gani. Mapurdo yung tindahan. So, the circadian rhythm characteristic but will continue to run even the absence na ng external stimuli or inputs, and it's about 24 hours in duration. But then can be reset or entrained to determine modified phase of period. Entrained or pwede siya i-interrupt and can compensate for temperature differences. Okay, so photoperiodism naman is the effect on the flowering of plant responses. So, like for example, kung short day plant siya, so ang dark phase is higher than the light. And take note sa mga equatorial countries like Philippines, we are tropical countries. We have fixed 12 hours day and 12 hours night. That's equals to 24 hours. So from pag sunrise na to 6 a.m., 6 p.m. po ang sunset. No? So, but then sometimes nga, kanina diba na ay longest day and but when you go to some other countries like sa Israel, sa oras maggabi dito, sir? Maggabi Diba? Mag-usab-usab. Sige, mag-adjust. Ang trabaho ninyo, time work, mag-adjust po. Ang work, pagkabalas na lang. Ang dili ang sa? Ay, ang uban. Ang Israel lang. Ay, yahay. Dato ang Israel, no? Sila equivalent sa money nila sa toa. 14 peso. Pero higher ang salary. Pare-pareho ra dire. Mas dako yun siya din. So, mudato, milyonaryo na kakaroon. Kaso gigas na nimo. So, yun, no? And pag long day naman siya, therefore, sa short day, for sa short day plan, kung mas taas ang light kaysa dark, therefore, dili siya ma-flower, vegetative yapon siya. No? But then, pag nagna siya light, then dark, short, dark, so, vegetative yapon siya. Kaya dili yun enough ang iyahang dark para maka-flower. Like the poinsettia, diba? Kung katong modified flower niya, mag-red baya siya ang leaves. Diba? Modified flower na siya. Diba mag-red na siya during December only, kaya nga naman, short day plant man na sila. And if ever, pero mag-flower na siya kung imong taguan sa dark. Walang nakalapot gani nga, And it needs 14 to 16 hours. Ha? Sa Bagyo na lang, Dre? Sa Bagyo na lang? So, photoperiodism is the response of plants to periods of lights and darkness. So, short day plants, such as chrysanthemum and poinsettias, will only flower when the days are short. Ito kasabot sa iyahang drawing. So, never mind. And for long day plants such as spinach and iris flower, and also nakalimot ko sa pangalan. They all flower when the days are long. So to differentiate short day between long day plants, pag vegetative lang siya kung ang short day plant, vegetative lang siya kung taas kayang life duration, no? But for a long day plant, taas ilang life duration, mag-flower siya. And for short day plants, pag this is the critical day length na 12 hours kunwari ni. And kung mulampas ang night of 12 hours, therefore ang short day plants will, but the long day plants will remain vegetative. And what will happen sa short day plants kung naputol ang iyahang day length? Night length. Flower gihapon ang long day plant kasi natugangan ang iyahang life duration. Pero ang short day plant will remain vegetative. So, sabta lang siya, no, between short day and long day plants. As long as for long day plants like the spinach, so ganun ginahimong bouquet. Kana sila, iris on. Kana sila, those are long day plants. I will only flower kung more than 12 hours ilang day. But with no interruption. Kasi kung interrupt siya, wala gihapon. No? And short day plants like chrysanthemum and poinsettia. Dapat more than 12 hours ilang night. So fighter cone is actually a pigment responsible sa photoperiodism, and it will absorb red light and activate a lot of signaling pathways in plants. An example is that Radiscantia, the growth response to gravity, di ba? Pa-downward siya. It will grow upward when young, but downward kung older. And this trunk also grow up away from gravity. Pero towards gravity na siya. And this was already, kung stimulating mo makahiya or the shy plant, no? It's the pulvinus in the motor order. How does cactus or desert plants survive? So how? Ang naiba lang nato, ang mga cactus mupuyo lang sila og kuan, diba? Ang desyerto, pero giunsa kaya nila? Okay, para saan? Para mangita og tubig and nutrients, and gamay lang mo nang nipis aga ng gamay sila og slender sila og leaves, and like cactus, oquetilius, and michubelias. Unsa na siya ang resurrection? Mamatay siya og kuan. Nakita na mo? Pangitawon na nato. Namoy na sir? Moni siya? Moni siya? Ay, wait lang. Ano siya? Ano siya? Wala siya pearl, no? So, pag butanganin mo siya o ka ng tubig, mabuhay na siya. Ang other term po niya is Rose of Jericho. Nag-isa rin na si Jericho. The Rose. Rose of Jeros. Ivy of Jeros. Char. Karon pa po ko nakadungog ani sir. Thank you very much for the knowledge and information. Asa na plant response. Dito siya sa katong desert plants. Gihapon. Dito siya. So naapot tayo ginatawag na carnivorous plants. No? And kita naman siguro mo aning ka ng Venus flytrap, pitcher plant, di ba? Sundews and butterworts. Using their leaves, they're going to eat the insect. No? Na sila chemical. Ano? This is the sundew, the pitcher plant. Makita po niyo sa Maragusan and the Venus flytrap. We also have parasitic plants, wherein they can harm their host organisms. The daughter plant, it has no chlorophyll and no food, kaya they will grow directly sa vascular tissue, like the mistletoe. So, sabi na kung ang mistletoe is parasite day siya sa conifer. So, this is the mistletoe and the daughter plant. So, we also have the epiphyte plants. These are plants that grow directly on bodies of other plants but will not harm the host plant. They will carry out photosynthesis instead, like the Spanish moss. It's not a moss, but is a bromeliad family. Nga nung itawag kong moss, no? Pastilan, murag peanut, which is not a nut, no? Coconut is not a nut. And strawberry is not a berry. So many orchids are epiphytes, and they like katong ano, diba? They will even help photosynthesis. So these are the Spanish moss. So common man kayo, diba? Nakita na to. And the orchids. And we have aquatic plants, of course. They have tissues. Pwede sila, di sila malumot because they have large air-filled spaces, no? Oxygen diffuses through these tissues and they will flow, they have floating seeds para ma-delay muna ang germination. And quickly after, they will germinate. And they have also growing shoot extending above water surface, like the cypress trees, bladderworts, and water lettuce. This one, water lettuce, water lilies, cypress, water lily. They are plant adaptation. So we have also soil tolerant plants. So ano nga yung term natin before sa soil tolerant plant? Sige, ito. So, itong salt tolerant plant. Sa term ato. You still remember? Katong sa classification. Ganito. So, review na po natin mamaya, ha? Itong different terms. Kasi ang plano na ako i-extract na lang itong term. Lagay bumalibog sa very long explanation na. Kaso nagbisyo-bisyo na po ko, karon. So, example are mangroves, shade scale, and salt brush. These are also examples of salt-tolerant plants, celosia, propomia, and opomia. And we can even use plants as chemical protection, like toxin to kill predator. Kating nicotine in tobacco, patay nga nung nagatabako mong ganito. That will kill insects and animals. And plant repel predators, milkweed tassels, which are bad for birds and insects. Okay, questions and clarification, you can also ask along the way. And last na lang before we proceed with plant breeding. Ano, wala lang.
Hi, Mr. Anderson. In this podcast, I'm going to talk about plant nutrition and transport. I'm going to use a number of terms you should be familiar with, like monocot, dicot, or ground tissue, vascular tissue. And if you're not sure what I'm talking about, make sure you go back and watch the previous podcast on plant structure. But before we get into that, we should talk about what plants need. In other words,
Hello,
what is
Pikes,
the
guys.
nutrition that plants count on? Well, it's the same.
Thank you, Madam Nova from Maragusan, Davao de Oro State College. Itong halophytes nga akong ginaingon na salt-tolerant plants are halophytes. So, review nato na ha, nakalimtan na nato. May pang top 1 na po ni si Madam Nova. Madam ha?
Thing is new. They need schnapps. And so basically what they need is carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. So where do they get the carbon? Well, unlike us, they're going to take carbon in the form of carbon dioxide. So that's going to come in through their leaves. They're going to absorb their water, so they're going to get their hydrogen and oxygen through their roots as well. But the other things like nitrogen, phosphorus, and sulfur, they're all going to take in through their roots. Now, how does that differ from animals? Well, we simply eat things like plants, or we eat things that eat plants, and so we can get all of those nutrients. So basically, we're kind of talking about the digestive, circulatory, and respiratory system all in one in relation to plants. And so basically, plants live in two worlds. They live in the world above ground, and that's the world of the shoots. And then they live in a world below ground, and that's in the world of the roots. And so basically, the one thing that they're taking in through their beads is carbon dioxide. And they're using that carbon dioxide to make sugars. And those sugars are going to make up the bulk of the plant, but they're also going to be used for energy. Now, as far as roots go, they need water. And so that water is going to flow in through their roots. It's going to move up through the xylem, and it's eventually going to evaporate through their leaves. And so they're taking that water. And the one thing that may puzzle you a little bit on this diagram is that they're also taking in oxygen. They're taking in oxygen through their roots. And you might think that seems a little odd. What do they need oxygen for? Well, remember, they do photosynthesis, and they're using photosynthesis to take in the energy of the sun and and convert that carbon dioxide into sugars. But they're going to use those sugars as well. So they do cellular respiration. What do you require for cellular respiration? You need sugars, and they're going to produce those through photosynthesis, but they also need oxygen. And so any part of a plant that's growing or requires energy is also going to require oxygen. Okay, let's start at the roots and then kind of work our way up through the shoots. I'm going to have examples here. These are the dicots and the monocots. So remember, a quintessential example of a dicot would be like a sunflower. And a monocot is going to be corn, or a dicot could be a dandelion, and a monocot could be grass. But basically what we're going to do is we're going to work our way up from the roots to the stems and then finally to the leaves. And you'll find that there are similarities between the two, but subtle differences. So if we start in the roots, what we're looking at here is a cross section. So imagine that a root looks kind of like this, and there's going to be root hairs coming out of it. Basically what we'll do is we'll just take a cross section, so we're coming right across like that, and that's why we have this circle. And so basically what do we have here? Well, there are three types of tissues. Remember, there's going to be an epidermis on the outside, so you can see the epidermis here. There's going to be ground tissue here in the middle. So in the monocot, same thing, epidermis on the outside, ground tissue right here. But you're going to have vascular tissue on the inside. So that vascular tissue is going to be made up of xylem and phloem. It's kind of hard to see with that color. Likewise here, we're going to have xylem and we're going to have phloem. Right outside. And so basically, if you think about it, in the roots, the vascular tissue, that's going to be the movement of water, the movement of nutrients, it's going to be centered in the middle of the root. Now, if you think about it, that doesn't make sense. How is the water going to get from the outside to the center? Well, every time we build new roots, you can see, this is roots actually in a potato, you'll see that they're actually starting at the bottom and then they're growing up from the center. And so it's kind of like a plumbing network where it's going to start on the outside, it's going to move to the inside, and then three-dimensionally it's going to go up in the plant. Now, if you look at the edge, some of these cells are modified into what are called root hairs. The function of that is to increase surface area. And a lot of these have fungus that are living in and around them, symbiotic fungi, that allow them to absorb even more nutrients. Now we're going to go above ground into the shoot system, and we're going to look at the stems. And so what you'll find is that we're still going to have the epidermis on the outside. See that? So here's the epidermis on the outside. It's going to have that cuticle on the outside of that. But what we start to see in the dicot is that the vascular tissue is moving from the center of the root towards the edge of the stem. The reason why is that eventually it has to flow out into the leaves. And so right here would be the xylem closer to the edge. We're going to have the phloem here. So this would be the xylem here, the phloem here, and the dicot. Likewise here, we're going to have the phloem here, the xylem, and then the phloem right here. But I always thought these look kind of like little monkey heads in the monocot. And then we're going to have the epidermis out here. What are all the other cells then? The other cells are going to be ground tissue. So that's going to be ground tissue as well. All right, let's keep going, because remember those stems are eventually going to bud off, and those are going to reach to the leaves. And so if we look at leaves, remember what makes a dicot is it's going to have this net-like venation. So you can see these nets that are branching off. Those nets, those veins are actually the vascular tissue. And so if we look at a cross section right here, the dicot, you can see that vascular tissue moving right through the middle. Likewise, if we go to a monocot, what makes them a monocot? Well, one of the characteristics is you're going to have all these parallel veins that are going through the leaf. And so you can see that those veins are vascular tissue that's moving right through the leaf. I try to kind of draw this through the picture. But both of them are going to have essentially the same characteristics when you get to the level of the leaves. Now we're taking a cross section of the leaf. We've got our epidermis on the top. We've got the waxy cuticle above that. We're going to have epidermis on the bottom. You can see right here that there's a stomata there. What's the stomata for? That's to take in carbon dioxide and also to lose water. Some of the other levels that we have in here is this. We're going to call this the Palisade Mesophyll. And then we're going to have this down here, which is going to be the spongy mesophyll. And so basically, what is that? And it's organized different in dicots and monocots, but basically, this Palisade Mesophyll, it's like Palisades, like a waterfall, is going to be an area where we have photosynthesis going on. So we're going to have light coming in, and that light is hitting chloroplasts, and that's where the photosynthesis is taking place. But what else do we need for photosynthesis? Well, we need that water. That's going to come through the vascular tissue. And then we need carbon dioxide. So the bottom part here, all of that space down here is for the carbon dioxide gas to diffuse in and can be used in photosynthesis. One quick thing I should mention, going way back down to the leaves again, our immune system kind of protects us from its environment, and also our skin is going to protect us, and it's going to allow things in that we want and things out that we don't. And so basically the way it works in the roots of a plant is a little bit different. And so imagine now we're down right in here, we're down in the root hairs like this. So how is water getting in? Water's flowing in through osmosis, so it's flowing in here to the middle, and remember it's going to go to the center of the root, and then it's going to work its way up. But we don't want to let just anything come in. And the way water flows is kind of odd. Some of the water is going to flow. So this is one cell and this is another cell. So some water is going to flow within the cell. So it'll go across a membrane. And it'll flow through these little pores between the cells called plasmodesmata. But some of it will actually move through the cell wall. So you can see this water right here will be moving through the cell wall. And it's never having to go across the cell membrane. And so basically what plants have is that, now let me do a quick cross section. So this is the epidermis, and this is the vascular tissue on the inside of a root. So the water is going to flow in like this. Basically, we don't want it to flow up in a plant unless we have had some say over what's coming in. And remember, what protects our cells, what allows certain things in and certain things out, that's going to be the cell membrane. And if you look at this water, since it's going all the way across here, we call that apoplastically. It's moving through the cell wall. It's never had to go across a cell membrane. And so basically what happens is you're going to have an endodermis here. This has a lining around it, and there's going to be a waxy cuticle called the Casparian Strip. And you can kind of see it right here. This is that Casparian Strip that's connecting all the cells. And basically what it does is it acts almost as, I don't know if you've seen The Lord of the Rings, but there's a scene where Gandalf puts his staff down and says, "You shall not pass." Basically what the Casparian Strip is doing is it's this strip that goes all the way across here, and it's basically forcing that water. It's made it all the way to the inside of the vascular tissue. It can't keep sneaking through the cell wall. It'll have to force its way into the cell, and it has to go symplastically down. So the function of the Casparian Strip is to force the water eventually to go across the membrane. And so we just have two things left to talk about, and that's basically how water moves up a plant, and then how sugar moves in a plant. We've talked about transpiration before, but I want you to think about it as a whole. In other words, there's going to be xylem, which are these vascular tissues that go all the way from the roots, all the way up, and the tree, it's going to go all the way up on the inside of the bark, all the way up to the leaves, and eventually evaporate. And so there's a connection all the way up from the roots to the leaves. But there's also something important you should know about water. Water is slightly charged, and that means that this right here, the oxygen is going to have a slightly negative charge, and the hydrogen is going to have a slightly positive charge. And so basically water will line up so that negative oxygen is attached to the positive hydrogen. And we call this bond right here a hydrogen bond. And so basically water is all connected through these hydrogen bonds. And so once one bit of water starts flowing up the tree, the other ones are going to follow. And we call that process, or we call that phenomenon cohesion. There'll also be some adhesion to the inside of that xylem. So two quick things you should know about xylem. It's dead at maturity. So these are cells that are now empty. They're not alive anymore. It's simply just a container or a vessel for the water to flow through. But basically, we go all the way up here to the leaf. Water is evaporating out. So the H2 is evaporating out. That water is connected all the way back through the vascular tissue, through the leaves, through the veins, all the way back down, through down here to the root hairs. And so we have a continuous pathway all the way up of water. And so every time one water molecule evaporates here in the leaf, that's going to pull this whole chain up. And so really xylem and the water is moved through a pulling process. What's generating all of that energy to pull it up? It's going to be the sun is pulling it all the way up. Okay, now last thing then is how does phloem work? Well, phloem is different. Phloem are cells. So here's one cell, here's another cell, here's another cell. They're empty. And so how do you be alive as a cell and be empty? Well, basically they'll have these companion cells that are going to have a nuclei, and they're connected to the phloem cells. And they're going to provide the metabolic support for phloem. But phloem is a living cell, and xylem, we said, is dead. They're going to be little connecting holes all the way through here, and same thing over here, connectors like that. And so basically, I like to think of it as push. And so phloem and sap and sugar is actually pushed around the tree. You might be thinking, how does that work? Well, let's look at this carrot. So we've got a carrot. It's planted underground. Let's say that it's the second year. This carrot happens to be coming back again. Well, if you think about it, where is the sugar going to be if this plant is just starting to grow? Well, more of the sugar is going to be in the carrot. And so if more of the sugar is going to be in the carrot, so we have sugar here, that sugar is going to flow out from an area of high sugar concentration down in the carrot to low sugar concentration here. We're also going to get flow of water in this direction, but basically the sugar is going to be pushed in this direction. Since there's more sugar down here, we call that the sugar source. And so phloem is actually going to be pushed from the source to the sink. Okay, so there's more sugar down here. Where's it going to go? Well, there's sugar all the way down here, so that sugar is going to start to flow up here. The leaves, it's early in the spring, we'll say, so they're starting to grow, so they're not making any sugar yet. That means there's no sugar here. And so we call this a place where there's no sugar, the sink. It's just like a sink, so the water flows from the source to the sink. Sugar does the same thing. So the sugar is going to flow up here, and then it's going to move into these cells. And so basically we get this flow of sugar like this. This is going to be early in the spring. But let's say the carrot starts to grow, and now we've got photosynthesis going on, and now we've got these leaves producing a bunch of sugar. Well, now this is the sugar source, so we're making a bunch of sugar here. That's going to push the sugar down here from an area of high sugar to low. That's going to be stored down here in the carrot, so that's going to be the state. And so basically what we get is this flow of sugar always from the source to the sink, but we have this flow of water all the way up the tree continuously as long as the sun shines. So that's basically how materials move around in a plant. Tells you a little bit about what they need, how they get it, and I hope that's it.
Okay, so this was already explained last week on flowing loading, yung shorter version ito. No? So now I hope you understand how plants take up fertilizers or nutrients from the soil and even water that goes up into the root, from the roots to the stems, through the flower and xylem, and being used by plants to the different sinks. No? So we have the so-called cohesion adhesion theory. Cohesion adhesion theory. So is all the explanation in PowerPoint presentation will be sent to you on that particular video. So in this cohesion and tension theory, ano yung naintindihan niyo? Nganong masuyop man yun sa tanom ang tubig o ang mga nutrients? Ang fertilizer ba masuyop dayon, ma-fertilizer lang alone? Kailangan ma-mix siya with water. So, kailangan to ang rain. Once ma-dissolve na siya sa soil with water, so what will happen? What particular part of the root will absorb this water and nutrients? The root hairs. And from the root hairs, how does it goes up? Unsa tong silent pool na ginatawag ni Anderson? Kung what kind of weather? Kung tag-ulan ba, mag-absorb pa siya? Di ba kuan mo itong naa siya adaptation kanang na eyelids na lagay na tulong ang mga tubig mo form? Adaptation. So it means nanubra na ang water pressure sa tanong. So, therefore, ang plants will absorb the nutrients and water through pressure. No? It's not even active transport, no? But pressure, water pressure. It's the natural movement ng ng water. Halimbawa, init kayo, transpiration, di ba? Mag-evaporate ang water from the leaves to the stomata, di ba? So, syempre, habang nag-evaporation, nag-transpiration, automatic tayo na nitrogen pressure. mo absorb dahil ay mo absorb dahil ang water with the nutrients inside from the roots to the vessels. Now, in terms of distribution naman ng photosynthase or nutrients, so what will happen, kapag i-explain ganina, from phloem to xylem. Ang xylem na, dito sa xylem na, ang photosynthesis, pwede siya ma-distribute. By the way, before mag-move ang water, natin ginatawag na cohesion. Meaning, ang capability or characteristics sa water nga mag-join together. Diba? Kung ibubuo nimo, dili na siya mabungkag. Diba? Pag bubuo nimo, ang sa mahitabo sa tubig, mag-isara po na siya ka, tulo, diba? As a whole. Now, if ever, say, sa isa ka tube, no, sa faucet, pag open mo sa faucet, nga naman mo, di ba, paanat doon ng faucet o paanat pa na siya pa-curve. Nga naman jud gamudagan man gid siya. Kay naa siya hydrogen bonds and tension, mupilit siya sa mga walls, no? Then because of the pressure. So inanak po na imagine na sa tanong. And it will take only how many minutes to do that for plants? Like, halimbawa, karon kung ang turgor pressure, na may mga 13 megapascal, may nanakunuhay, no? Na if ever ang pressure is kusog, pila ka minutes na maabot, depende po sa height sa tanong, no? But for example, 1 feet lang imuhang, 1 foot lang imuhang tanong. So pila ka minutes lang maabot ba dayon ang water sa lids? Pila day ka oras adlaw? Depende sa pressure, diba? It will only take, tanaw ninyo, estimate, minutes and hours, diba? Yeah, that's why kung mag-spray mo, when is the best time to spray? Kanang walay ulam, of course, diba? Kaya nga, no, para ma-absorb, no? And halimbawa, ni ulan siya, kanusan mo, ni ulan siya pagkawin mo spray, pila ka oras dapat ang nilabay para makaingon na ka, ay safe na to, na-absorb na to. 2 hours to. Very good kayo sila, sir. Of course, mga expert na actually. Board exam na lang ang ginahulat. No? So, estimate in-ana din siya. No? Na within hours and minutes lang. Depende of course sa kadako sa tree of tanong. Kung alangan na magkuan kung highest tree in the world. Say tallest tree in the world sa man. Mga 100 meters, diba? 1,000. What is the tallest trees in the world? Katong ganina, unsa man to? Mga redwoods, no, sa California. Diba? Sa kitawanan nila, scientific name. Kalimut ko. Redwood. Madam Nova, call a friend. So, yeah. So, and then, how would you know nga ang photosynthesis na tumato dito sa flowers? Anong dili sa leaves? Anong dili dito sa fruits? And ang movement niya, na-mention si Anderson nga apoplastic, symplastic, diba? Kasi, kasi pwede siya through the cell, that's aposymplastic. Kaya mong friend, plastic jod. Apoplastic siya kung sa cellular. Pag sa cytoplasm, symplastic. Pero naputay 2 parts of movement from cell to cell. We have the active and the passive. Active kung kailangan din ng energy. Kasi wala yung di madutlan ang kanang pressure or natural hydrogen bonds. So kailangan din ng energy. Kailangan ng ATP mo. Gamit siya ATP para dadon niya. And in that time, for example, is when ang sink nato is the growing tips of flowers and fruits. No? Kung mag-allocate na taog photosynthese para mamulak na siya or mamunga na siya. Pag passive lang, no need of energy, no? Musunod lang siya atong apoplastic o symplastic ways of diffusion. So we all know about diffusion. It's the movement of water from higher, lower concentration. Wala dara actually ang ginayaw-yaw na adari sa. I-send na ko ni sa inyo but hindi na ako i-discuss because I already shown you the video. And our last discussion for this afternoon is about GMO and plant breeding and genetics. So ako ni isend mamaya. Sorry ha, kahit na ako naka-use up ko siya. Okay. Ayan. So, dili na ako na-explain ha. I have a lot of details also here. But if you have question or clarification, muna akong ginamina adaptation. So, I will just send this to you since we don't have much time already. It's a beautiful explanation and I hope sa simple lang to nga version na to na sabta na ninyo. So our next is before I will start plant breeding and genetics, I would like to entice your mind muna first on biotechnology and its status. Although dugay na kayo niya siyang overview of biotechnology concepts and application, dugay na kayo nino sa circa na presentation. I guess 2011 pa ni. But there are also a lot, but kani is very specific in the Philippines, and that's why it's nice. No, the historical development, biotechnology, genetic engineering, and application, as well as the global status. So, as I have said, nung una, we started hunting and gathering called pastoralism. So, the man as old as they existed as food gatherers and hunters, or Homo sapiens. Homo sapiens man ta tanan direct, no, dili man ta Homo habilis ba ang saga. Domestication of animals and selection of plants began mga 8,000 before Christ. And then meron na tayong parang genetic engineering or modification actually started when we started making wine and baking leavened bread, kasi doon na din nag-start ang fermentation by the Egyptians around 4,000 before Christ. And in 1760s, the age of industrial revolution, wherein nanatay mga pandaro, mechanical implement and machineries. In 1860, we started plant breeding or pollination of plants. In 1960 to 1970, it's actually the period of agricultural advance known as the Green Revolution. Ano kaya? Kasi nagagamit na taong mga high-yielding varieties, na natay fertilizers na lalaman, and irrigation, pesticides. So, hoping that we had strengthened food security. And then in 1953, Watson, Crick, and Wilkins Franklin have found out the structure of DNA using microscopic force. And in 1973, na-discover na ang recombinant DNA by Cohesion, and this is the birth of modern biotechnology. So 1970s. And a decade after, 1983, insulin was first produced as the first product of genetic engineering. So, ang mga diabetic na naitambal karon because of this. And the first successful field trial of the BT cotton was on 1990. And the Human Genome Project was established in 1990. So, lahat na sequence na itong genome. And in 1994, Flavor Sour Tomato was already approved by FDA as the first biotech food crop. Kasi yung first product is insulin. But the biotech crop is like Flavor Sour Tomato. And in 1996, we are already commercially growing mga 1.7 million hectares of biotech crops in Argentina, Australia, Canada, China, Mexico, and US. In the following year, 1997, ipanganak si Dolly, no, the first cloned animal. And in 2000, already 44 million hectares in 13 countries. So, ang first genome naman sa plants was developed in Arabidopsis thaliana. And until from here on, nag-start na actually ang golden rice, no? And you remember until coffee break. Gusto ba daw mag-coffee break? Of course. Especially pagkapi sa daw, break sa ta. Oo, and that's courtesy of the birthday boy last week, si Sir JP Matugina. And so, naipa-burger. Pagbalik ka, paitong pan ka. Sa'yo mga pisa ta. So, 20 minutes break, Shara. Actually, abit na tamahuman, no? Kasi we will talk about plant-breeding and genetics na lang. And, magkuha na po tayo sa ka-topic, which is plant propagation next week na nato. Right? Next week, ugma day. Unsa? Sa tanaw ni mo. Very good, guys. Giant Cicada ay katong redwood. Thank you, Madam Nova. Sureness na yun ganyan itong top 10 ba? Visit kayo ko, nag-review ko. Look. Ha? No. Okay, so to continue on the different GMO crops. As I have said last time, golden rice, if you remember on the future of farming, sa ating history na video, was already about to be distributed na, no? So golden rice is having supplement with vitamin A, no? Kaya siya gold ang color. And this is para ma-treat yung vitamin A deficiency. So next, we have, as of 2009 pa lang, since 2009, 25 countries na yung merong biotech crops all over the world in 134 million hectares. So yan, so pa-increase ng pa-increase. And now totally, we have adopted this GMO crops already. No, diba? You don't even know that you are already eating kanang GMO corn, diba? BT corn. No? Maski pag dili mo mukhaon ana, and then ang pakaon sa inyong baboy is BT corn man, diba? Murag yapong istoryaha. Okay, so speaking of biotechnology, this includes animals, yung development of vaccines, plants, microorganisms, biocontrol against pests and diseases, mga recombinant DNA, monoclonal body production, embryo transfer, fermentation, protoplast fusion, plant tissue culture, and biofertilizers. Those are products, all products or scope of biotechnology. So when we say bio means life, and technology is a technique wherein we use as a procedure to develop new products. So this use living organism or parts of them para ma-modify, ma-improve natin ng animals and microorganisms. So it has a lot of discipline, no, involved, like genetics, microbiome, biochem, computer science, molecular bio, food processing, food tech, food science, breeding, and mechanical engineering. So in the DNA or genetic engineering, these are technologies that very artificial move functional genes across special boundaries para makaproduce tayo ng novel organism. So in the future, we're going to produce na mga resistant crops, high-yielding crops, no? Yung mga ganon. So these are just the procedure in capsule, in genetic engineering. How does they develop from DNA, RNA to protein, transcription and translation. So una piliin yung clone gene of interest and introduce it to the cell, and the cell will express the gene, and proteins are then produced, then ma-express na yung trait. So it will took time in a traditional plant breeding technique. It could take up to 20 to 50 years, but with the kind of technology that we have in genetic engineering, this will only cost us 2 years for our vaccine, diba? Or less than a year. Less than a year, guys. In less than a year or 1 year, na tayo vaccine, diba? So all we need is a vector, a host cell, and the technique or way to introduce the gene into the cell. Sometimes it's in the form of a gun, no, gene gun. And of course, we need time and money. That's very expensive. And the clone of gene of interest, the problem. So breeding with precision accuracy through manipulation of genes. Why and when to use if yung ating gustong trait is not present in the commodity or crop, and if the trait is very difficult to improve using conventional methods of traditional breeding, at para ma-improve na rin yung mga trait that will take time, especially ito ng mga persistent resistance to pests and diseases. Those are very important and high yielding. Kasi nga nung magkuhan man siya ka, mag-antos man siya ka, sa gagmay kita na breeds or mga varieties kung pwede naman katong high yielding na. So overall, this is the process, no example from the vector or the marker gene, and it transferred siya sa parang bacteria or cell, and then yun yung through the cell will generate or multiply or gene transfer through plant tissue culture, then will be selected again and will be tested in the field. So later on, you can have the new variety. So what are the application? Unang-una, bioprocess technology. So sooner we can produce essential primary metabolites like acetic acid and so on, and mga industrial useful enzymes, lactases, monoclonal antibodies, vaccines, and novel products. And in the environment naman, waste treatment, no? Dali na mapa-degrade ang mga plastic, for example, pollution control, for example, mga monitoring and detection ng mga contaminants, and removing toxic waste, or we call it bioremediation. And also recovery of metals from mining waste and low-grade areas, we call them phytoremediation or accumulation. So it's not only for the crops, for the food, but also for the environment. And for genetically engineered plants, for golden rice, for example, for improved nutrition, vitamin A, and resistant to pests and diseases like the BT corn and papaya ring spot virus resistant, PRSV. So we also have the improved yield and stress tolerance, drought tolerant crops in aluminum and manganese tolerance, and also post-harvest characteristic of flowers of tomato, as I have mentioned earlier, and in the cut flower industry, nagdali lang ko, tissue culture technology in banana orchids and biocon for like the rhizobium, the nitrogen fixation, and bioherbicide. These are all application or products of biotechnology. And in animals naman, we have the super mouse, or it's the first GE animal with growth hormone gene. And in livestock, commercial, improved na yung quality ng meat, yung egg, yung wool quality and quantity. And we have already vaccines, also bovine growth hormones or somatotropin or BST for bovine. It's for the animal agriculture, injected to cows for increased milk production. And of course, vaccine for animals. And for healthcare, also for humans, we have hepatitis B vaccine, and we also have our COVID vaccine now, insulin for diabetes, somatostatin for growth hormone, interferons for cancer and virus diseases. And therapy or gene therapy, the treatment of genetic disorders and disease cells using in the future papala, in the near future, but this has been done already. I-manipulate yung gene para ma-cure yung disease, for instance, cancer. So I guess yun lang, no, and this is just to show you the global area of biotech. But this date is up to 2009 pa lang. And the trend is increasing. So any question regarding in the Philippines, global pala pa ito. So for soybean, cotton, maize, and canola, very high na ang is increasing na ang biotech. And also adoption in Visayas, Luzon, and Mindanao, highest ang Luzon. So there in the Philippines naman, we already have approved GMO for BT, RR, or ground-up ready na corn and soybean and ano pa yan? BT or Bacillus thuringiensis, both BT and RR ready na mga plants. And there are already 49 for direct use na mga GMO. Ayan. So ito yung estimated na ating Philippine GMO. And as of now, in the Philippines, we have the PRSV-resistant papaya, the BT eggplant, and BT corn as well, and BT copan, the late ripening papaya, golden rice. Kasi nasa atin ang International Rice Research Institute, diba? And it is this golden rice is being done in Erie. It's in Los Baños, Laguna. And the virus-resistant sweet potato. Okay, so questions so far. So we have again the IPB, the Philippine Rice Research Cotton Development Authority, and the Visayas State University. Looking forward for initiatives on biotechnology. So mga pangutana, before we proceed with our presentation on plant breeding and genetics. Ha? Apil na? Sa ako ang time, wala ko na question na biotechnology, but my God, that was 2011 pa. So, in previous time, no, na naka-mention, na ko nadugog na nanay mga GMO. So, short lang man naman siya, just to explain about update on GMO. So, pardon, kasi, ano kayo? So I'm just gonna use my old presentation on plant breeding. I mean genetics. That was
Good afternoon, everyone, and welcome to your subject. Lesson 22, agriculture. He said that physical traits are responsible for the. So, it is actually called Mendelian genetics because it was the one who is the founder of genetics. And he, from 1822 to 1884, was responsible for the laws governing inheritance traits in his country. So Rico Mendel was an Australian who studied the inheritance of traits in plants and developed the law of inheritance. Gregor Johann Mendel was born between 1856 and 1863, and cultivated and tested some 28,000 plants. We found that the plant offspring retain traits of appearance. So this is the actual type of the good warming vertical garden in Czech Republic. So Mendel stated that physical traits are inherited as particles known as the particulate inheritance, although he did not know that the particles were actually traits. And there are various genetic terminology that need to be defined, like for instance, we have trait, it is any characteristic that can be passed from parent to offspring, is heredity, and another one is heredity is the passing of traits from parent to offspring. So there are types of genetic crosses, the monohybrid cross, or the cross involving a single parent, like the flower color, and the hybrid cross involving two square is used to help solve genetic problems. So as you can see, this is the male gamete, separate the male parents and the female gametes. And if you, in the Punnett square, it's just pairing them on the square, as shown in the diagram. So the P for the P, small p, and another small p from the female gamete is equal to the same as the double capital P and so to make the Punnett square, this will allow geneticists to predict the possible genotype and phenotype of offspring. In this example, the parents are the heterozygote for yellow pea allele. Y is to Y. So this is the parent one, the heterozygote Y, and the parent two Y and Y. So to make the grid like this and place the alleles of the gamete of one parent along the top of the grid and the other parent along the end of the grid. Then you fill the grid to find the parent alleles inside the boxes. The letters show the genotype of the offspring. So we pair two capital Y and then one from the female, the parent one capital and one small y. And then in the second pairing is one capital Y and one small y. And the fourth pairing is one small y plus another small y is two small y. So the genotype ratio is 1 is 1 is to 1 is to 2 and 1. Meaning there is one double Y combination and there is two big letter Y and small letter Y combination. One small letter Y. So you see the law of dominance to determine the phenotype and the phenotype ratio of the offspring that you have here. The phenotype ratio is 3:1, meaning there are 3 yellow peas to green peas. In the capital Y stands for the yellow pea, the small y stands for the green pea, and because the capital P is more dominant, the capital Y is more dominant, so it will color yellow, while there is only one green. So the designer genes, known as alleles, are formed to genes which can be dominant and recessive. The dominant is the stronger of two genes expressed in the hybrid, represented by capital letter R. So genotype is a gene combination for a trait like a double capital R, one capital R and R, small r, and while the phenotype is the physical feature resulting from a gene combination. So the genotype and phenotype in flowers, for example, we have R for red flower, the capital R is a yellow flower. The all genes occur in pairs, so there are two alleles affect characteristic of the combination. R for genotype, there could be one double capital R, one capital R and one small r for double R. So the phenotype for the capital R is red, double the G capital R is red. The second one is first capital R with small r still red because it's dominant. And the color for the double letter R. So talking about genotype, it can be homozygous or heterozygous. I'm sorry for the slide. You can always go back and check this one later on. So I cannot maybe if I open this one to the slide, I can show you the whole text in the slide. So while waiting, we've done this one. So another type of genotype are homozygous and heterozygous. So homozygous are gene combinations with two dominant or two-percentage genes, so meaning there are double capital R or double single R, small letter R. They are supposed to be pure. While the heterozygous genotype are genes with a combination of small and the big letter R or dominant and recessive genes. So we are now here on the genotype. So in here, the genes that interact and in both genes and environment will determine the characteristic of a particular living organism, including human beings. So they say that 90% is genetic. The actual identity of a person is 90% genetics and 10% environment. All in all, that determines your quality as a human being and as an organism, whether plant or, and we know that human beings belong to the plant kingdom. So this is exactly what Mendel's D-plant experiment. So he used actually peas because, or the Pison Katibu, because it can be grown in a small area and produce lots of offspring and also produce pure plants that allow itself pollinate in several generations. And they can also be artificially pollinated. So I'm sorry for this, but some of the slides actually showing well. So the reproduction and flowering plants is involving actually we have many here. This one is the biology lesson before, but this is the reproduction of the plant container, the male reproduction organ, and the pistil and the ovary is the reproduction. So the pollen contains sperm produced by the stamen. These are the pollen from the stamen, and the ovary contains the egg inside the flower, and then the pollen carries sperm to the egg for self-fertilization, and then self-fertilization can occur in the same flower. And if it's not, it's therefore cross-fertilization can occur between flowers, especially for those on incomplete flower. Incomplete flower meaning they don't have the full male either one of the male or the female. So just like the monoecious plant, they only have they have both separate male and female flowers. So they are cross-fertilized or it's not So the Mendel's experimental method used hand pollinated flowers using the net brush and using the paintbrush. He could stick the stamens to prevent self-pollination. Covered each flower with a cloth like that, and he traced the thread through the several generations. So the Mendel used traits by allowing plants to self-pollinate for several generations. So I should show the picture. And then after that, there were actually 8 different traits that was produced. In terms of seed shape, there were round or wrinkled, which is representing the capital R, and wrinkled is small letter R. In terms of color, yellow is capital Y and green is Y. And for bud shape, green is capital S and small letter S for wrinkled. And bud color, green for capital G and yellow for G. For the seed coat color, we have gray or capital G and white. And for the flower position, the axial is capital A and the terminal is small letter E. And for the plant height, capital letter E or short small letter E. And for flower color, we have purple, capital B or white. Now let's take a look at the combination here. In pictures, these are the pigment therapy studied by Xylem. We have wrinkled, yellow, green, inflated, wrinkled, green, and yellow. We also have purple, white, and flower position of stem lentil, and more. So those are the some of the characteristics. So based on the experimental result, the ratio of dominant to recessive in Mendel's plants are, we have dominant traits are the smooth, it yellow, it inflated, green, purple, flower, and stem. Other recessive traits were wrinkled, meaning there is small letter R or Y. Green seed, wrinkled pod, yellow pod, white flower, flower seed, bark, and average or ratio of green is 3:1, meaning these are the dominant traits. They dominate. They always this is the recessive trait is in the combination. So the ratio of dominant to recessive in the second generation, filial generation is actually 2.96 is to 96.96 to 1, which is 5,474 smooth and 1,850 so on. So usually the ratio of the dominant B is to 1. So data observation match the theoretical ratio. So the theoretical or expected ratio of the plant producing around or wrinkled is 3:1 wrinkle. So Mendel's observed ratio was 2.96. So there is like 1:1, 2.96:1. So the discrepancy is, well, maybe because I statistical error. The larger the sample, the more nearly the result approximate to the theoretical ratio. But it's actually when you conduct experiment, it's very important to have a large population. You can estimate of the theoretical population. Now we have a so-called generation graph. We have parental or P1 generation, the parental generation of the experiment. While the F1 generation, the first generation offspring in a breeding experiment or the first filial generation from breeding individuals to P1 generation. Well, the second or F2 generation, second generation offspring in a breeding experiment which is the second fluvial generation from breeding from the F1 generation. So following the generation, so it starts with the parent generation and the first fluvial generation and the second fluvial generation. It will cross the two-tier plants like the dominant T and the recessive T, so they all result in hybrids with heterozygous dominant and recessive traits. So if another on the second generation, then we will cross the two hybrids, they you get 3 tall and 1 short, as shown on here. So let's talk about the monohybrid process. Monohybrid process is, for example, the trait is seed-shaped. So there are 2 alleles, brown and wrinkled, as represented by T, there are R and one of the R. So when you cross the brown seed with the root of seed, so this using the Punnett square, so you will pair the first big letter R with a small letter R, making the capital or the big letter to come first, despite of their arrangement. And pairing again is R and R, the second pairing is first letter combination of big letter R and R. And the next combination is again left big letter R and letter R. The third combination is the big letter R and letter R. All in all, they actually have the same combination. We're talking of monotype one. So the genotype is all big letter R and letter R. The phenotype are all brown. They start with they have all the capital for the first big letter R, which is the brown one in the dominant allele. So we have genetic ratio as alike, all alike, phenotype ratio as all alike. So they are all actually. Yeah. So for the monohybrid, for a review of the monohybrid cross, homozygous dominant times homozygous recessive, the result of being all heterozygous hybrid. Well, offspring and the offspring is called the first filial generation, the genotypic and phenotypic relation of life. Next, for the second layer monohybrid cross, again, if you are going to cross the first filial monohybrid cross, meaning they have the same trait and allele. So we have again combination of the last cycle. So you will have the first pairing, capital R, the next pairing is between small and capital R, and then R is a small capital big letter R. The last pairing is small R. So the genotype is we have first all capital R in combination of the capital R, letter R, or the grammatical, so all being all parts all letter R. So the phenotype that will come out is brown and with a ratio of 1:2 and 1. Well, the phenotypic ratio is 3:1. It means there are 3 brown, 1 and 4 wrinkles, or 1 wrinkle. So, as a review, in a heterozygote and with from the option would be 25% homozygous R and RR and 50% heterozygote combination of P letter R and 25% homozygous recessive trait of small letter R up to second cell generation. So the phenotypic ratio is 1:1. And the phenotypic ratio is 3:2. So what could the piece design? So as shown on the figure, some of these pieces have a full texture, while others are plain. Now, Mendel then crossed a pure and a hybrid from this second to generation known as the F2 test cross. So there were key possible test crosses, the homozygous hybrid and the homozygous recessive kinds of hybrid. Let's say again, homozygous they have the same dominant capital R and homozygous recessive is the same small letter R. So in the F1 hybrid cross with a seed shape, and so we have between round seeds and another round seed, but hetero.
Okay, sorry to cut it down. Kasi we're running out of time. And hinay kay siyang bayhana siya, no? Saray siya. Wait lang ha. Tamo ka bang bayhana, hinay kay siya. So i-fast track na to. So I hope you have a bit, I know review na lang man po ang genetics, diba? So you should have able to understand what's a Punnett square. Muna para walang model para i-pair nato ang mga kanang outcomes sa itong debris, no? Like for example, no, kung i-translate nato Sir Lance, ikaw man ang tig-breed diri iro good. Sige, in application sa imuhang iro. Kung halimbawa, parin P1 ka, no? First parent generation. Mga unsa na nimo mga iro karon? Kapila na nga generation? Ha? Second pa lang. How did you know? Like, naa kay breed sa iro nga first, original gid siya nga lahi. Sige, example ni daw. Unsa? Eagle? So kung beagle and beagle, unsa man na siya? Ha? Kung the same breed lang. Inbreeding siya, diba? So advisable ba na sa mga iro? Line breeding gani. So both parents, no? So kasi kani sa P1 natay first filial generation and second filial generation. Monohybrid cross na siya, meaning isa ka character. isa ka kanang genotype. So, genotype versus phenotype. Of course, you should know the difference. So, when we say genotype, ano na siya? Ang iyahang genetic combination. Katong RR, small r, capital R, and ang iyahang phenotypic physical characteristic kung unsay makita nimo, no? Regardless kung unsay combination niya. Nawala na po. Anyway, so kasi kung halimbawa, ano siya like so itong kusog na recessive and dominant, sorry. Kung dominant siya, like kung ang dominance is represented by capital R siya, di ba? O, whether, so kaning pila ka dominant dere? Pila? Upat, kay upat ang capital letter, right? Therefore, they all look like the same or different. So, ang capital R dere are the round seeds. So, therefore, tanang, ang phenotypic niya, characteristic tanan, round. Okay. So, another one is first and second. So, pag i-mix naman nimo ang small rr and double rr, okay, what will happen? And sa Punnett square, again, imuha lang yun na siyang itunong-tunong mo nang, and then first, big letter, and then small letter. So, pag tunong-tunong ni mo, ana, that's why Punnett square siya. Imong genotype, ana, is dalawa. Diba? Yeah. And the phenotypic, therefore, na kay round, na kay wrinkled. So, ang ratio niya, therefore, kasi 2 is to 2. So, 1 is to 1. Nasabtan pa ninyo? Tama ba lang pag-explain? 2 is to 2, so 1 is to 1. Dili ba? I mean, ang ratio niya, equal ra. No? Pareho ra. Na ay duha ka R, na ay duha ka round, na po duha ka wrinkle. Kani-review na lang mani, which is already, So, Mendel's law. So, before we proceed with Mendel's law, let's go back to Lance. So, unsa naman to Lance? In application naman sa imong ero, what did you do with two breeds? Crossbreed siya. Line breeding siya. Pareha sila parents. Pareha sila breed. So, unsay offspring nimo? The same phenotypic ratio. The same characteristic, the same breed. Magkapareho gid ang ero, no? Para sa mga kabaw, pareho sila tanan, di ba? Diba? And then, ang anak niya, kamukha niya o na-change sa color. Ayaw gani ko, ako pa ginabinuangan ni mo, Lance? Ang sabi, dili ba? Wala kasi ko asa naman to akong malusna? Wala po, ayan. Kung kakabit, dali, Sir Lance, magpunta kaya ka. Sir Lance, sulat yung breed na rin. Sulat yung iro na rin. Breed ka, kung saan ka breed? Spell mo. Eagle? Kung ba palo? Anak? Dali yan, dali ba? So, unsay color ang in terms of hair color? Tricolor. Unsay tricolor? Sa color no. White. Pareho sila color? Orange. Dadami klase na color na white. Ano ba ang color yan? Color lang mo. Yan na. Brown, white, and black. Ayaw kayo ba yung ginawa ni Sir Lance? Nga na ikaw nag-print sa hero mo. Hoy! Ilan hero? Oh, 15K lang iyang hero, ha? Oh. Zaman. Oy, asthmatic ko, sorry. Ang sa first lang ako. Bigyan na color. Anaran na magawa. Thank you lang. Magaganda yung ganda ko na ito. Tayo na. Rot ka na sa ilulo. Sa lola mo. Ganda siya ako. Yan. For animals yan. Tamdan. Hindi naman bitala. Like for example, sige nga, kasi gusto mag-share. Dara, si ano. Oh, ikaw, Argel, kaganina, sige ka gano'n. Kung sa'yo mga lahi ninyo, sa imong parents. Tribo. Australian na. Garon. Kapila ka, Jel. Pila mo mag-siblings. Magulangan ka. Ikaw po lang pinakagwapo. Ang pinaka-tangkad. So, ang imong mga magpapa are, Bisaya o pure sila, duha Mandaya. So, original Mandaya mo, 6 ka buok. Walay walay mixed breed. Wala. So, original siya. So, but first, so, ikaw po ang pinakatangkad sa tanan. Naikalahian within the siblings. Ano nakatawa mo? O pare-pareho ra mag-height. Hala, alam na, no? Hastang malasa po nimo. Imong lolo, parehara pa nimo height. Lolo, lola. Wala. Silingan juga. Cross-pollination. So, thank you, Jel, for sharing your genetic makeup. So, kung nakay kasuko, Pero ikaw gyapon ang pinakagwapo sa inyo. Dili oy mas tangkad daw siya. Siya ra daw ang ano. Tangkad naman ka, just like height mo. Ha? 5'3 plus 5 inches pa yun na. Joke ra. So, going back sa ating Mendel's laws. So, there are laws no to be considered in genetics. So, unang-una, inheritable factors and genes are responsible sa lahat ng ating characteristics. So kanina na nakita ninyo how to do the Punnett square. It plants to siya. And in animals naman, it also it is still the same. I will show you tomorrow how does it applies to plants. This time this is for plants, for animals naman. No, kasi sa animals you have to consider the mga inbreeding, mixed breeding, kasi na for animals, iba-iba ang kanang inbreeding is not good, no? Sa animal production. And even sa tao, they believe, but it's not really, it's not only siguro sa mga, it's because sa mga royal family, di ba? Alam naman natin about royal family. They only marry their cousins of the same, kaya they have the so-called disease in the end of this, yung katong blood clotting factor na pag masama sila, hindi sila maayo, diba? So, ma-mention mo na mamaya. So, all are inheritable and although dili siya physical, like kanina, makita man ninyo ang recessive and dominant. So, although dili siya dominant, but you still have the genes, and it will be passed from one generation to generations in your family. So phenotype is based on genotype, of course, and each trace has 2 genes from your mother and from your father, of course, alangan, no? Sa silingan. So through breeding individual, or yung homozygote, yung both alleles are the same, or the characters. So, the law of dominance. So, of course, we understand already na kung dominant trait siya, yun siya? Makita siya sa phenotypic ratio. And, as shown in here, no, na-explain naman nato, na ang dominant there is the yellow. Pastelan. Sorry, nasuko na siguro ng mga online sa ko. Okay, so I have just shown you here kanina about the law of dominance, so Mendel's law. So sa law of dominance, which is based sa kanang still based on genotypic ratio. So if like ang dominant is represented by capital letter, therefore kung unsay dominant mo dito yung gawas tanan. Like for example, imong mama magpapa have black hairs, and then kamo mga anak black hairs po, no? Then if naamay mo gawas isa nga kanang brown hair siya, ibig sabihin sa pasabutan na Sir Pundal, kung halimbawa nalahi mong tagway sa imong mga igsoon sa imong papa sa pasabutan na lahi mong papa, no? Yes, from the ancestors. Charge na lang nato. Charge to ancestors, no? Mong lolo o lola. Naku, ginayin mo. Naun sa day mo. But 90% na po ng DNA test. Kasi, dili po accurate na na. And for the law of segregation, ano yun? Siyempre, pag mix kayo, ano na naman, di na nila siya mag-sugya po. Wala kasi din ako yan. Si W sa laani ba? Okay. Nag-resume na slideshow. What happened? Ano yun na siya mga screen 1 and 2? Dili po siya. Stop share na lang kami. Okay. Screen 1.
Kung dati, kung nana, di naman sharing mo sa screen 2 mo.
Screen to this. Ako ay nalalago pa sa iyo. Okay, kasi okay na. Kasi naka-slide share ko kanina. Okay, so we are now on the law of segregation. Okay, ayun na naman siya. Okay, in the law of segregation, so what does it mean? The two alleles from different parents, from your mother and father or exon sperm, will combine during fertilization. So, masegregate na siya, one from each. And to apply that, ito yung sa Punnett square, no? Yun lahat will be distributed. Like kanang, ang tanong nimo, ang imuhang animals or your parents are both tall and short. So, ang malamang, tulo sa inyo are tall and one is short. Pero kung tulo ra pud mo anak, nara pud ani nga combination. And kasi that's segregation. And we also have the so-called law of independent assortment, meaning the alleles of different traits will be distributed to sexuals and offspring independent of one another. So, pareho ba mo, each of the siblings, each of the plants, each of these animals does have the same alleles characteristics? Lahi-lahi. But then there, yes. No, kasi because it is assorted already. And after assort, but then naatay ginatawag na katong unsa man whatever na mix sa iya nga recessive, although wala physically nagawas, pero later on sa next generation, magawas na siya. Okay, and then using the dihybrid cross, of course. No? Wherein, kung dalawa ang different i-cross nimo, two different characteristic ang imong i-cross, therefore, in the dihybrid cross na kay upat 4 ka combination, diba? 1, 2, 3. Nag-quiz na siya. So the answer is 4 combination. So I will just focus on the nag-quiz na dun siya. Dihybrid cross. So this is what I'm talking about. Kung ang dihybrid cross nimo nag-mix na, so upat ka possible characteristic or gamete combination. So it has been already discussed last time, then this will be shown in this Punnett square. Wherein meron tayong how many rounds? 9. And brown green, 3 wrinkled yellow, and green wrinkled white. So 4 possible phenotypic ratio. Okay, so in the next meeting, you will know kani test cross naman ni siya. So again, monohybrid cross will only involve one character, but one allele, but dihybrid, 2 different characteristics. So to summarize our Mendel's law of dominance. So for monohybrid cross between tall and short, you're going to have all 100% na tall. But for the law of segregation sa dihybrid cross, you are going to have 75% na tall and 25% short. And in the independent, still monohybrid, sorry. Segregation is monohybrid but second generation. And this independent assortment is for dihybrid cross. Wherein, kay 4 ratios. So I guess yun lang, no? The next topic is incomplete dominance and incomplete dominance and co-dominance. So what does it mean? When you say incomplete dominance, these are appearance between the phenotypes of 2 parental varieties. No? And like red flower and white flower, but these are both on the flower. So incomplete siya, dominance, because nag-rely siya sa phenotypes sa mga varieties and somewhat in between lang siya. So, but it's still the same, no? In the first filial generation, it all produced the pink heterozygote. So, for example, in this one, between white and red, no? So, ang combination niya may 5, 2, 4, 5, and 1 white. And with this combination, in the second generation, so ang makukuha niya is white, pink, red, pink, and white. So, incomplete dominance siya. And codominance when 2 alleles are expressed. So, na siya both multiple alleles and heterozygote individuals. So, these are applicable for blood type A, B, ABO. So, ang pwede for type A, and type O. Ayan. So, if i-combine mo siya sa homozygous male and heterozygous female with type B and type A, of course, using the Punnett square, mo hang blood type 1 half, type 1A and 1B. So that's to show you the codominance. So, if a boy has a blood type O and his sister has blood type AB, so what are the genotypes and phenotypes of their parents? AB. So the answer is A and B. Very good. So we have also called sex-linked traits. Now we're in the sex chromosomes, we all know that pag babae ka, you have double X, diba? Pag lalaki ka, you have both the X and Y. Kaya na tendency nga, karon lalaki ka, pero later on, diba? So kasi this is to show you the, for example, the eye color input lies, no? This is called controlled by the sex chromosomes. So this is the X double X for female and X and Y for male. And using again the Punnett square with eye color, red eye male and white eye male. So you're going to have 50% red eye and 50% white eye. So that's for, and also yung sinabi ko na female carriers like the hemophilia, no, katong di wala sila blood clotting. So it only runs with the royal blood family kasi both, ano lang man, ilahang ginamarry, ilahang man within the family, within their cousins lang para ma-preserve ang ilahang family power and also kingdom and their wealth, no? So the women here are carriers only and the men are the one which expresses the characteristic. Okay, so the same pa rin using the Punnett square. So some, I guess yun lang, no? About hemophilia, that's for, these are all for, ano na, for medyo, ano na siya, for humans. So questions, clarifications regarding, ano, genetics. So I'm sorry, we don't have much time for animal breeding. So we'll continue and also plant propagation on tomorrow. Tangutana, Sir Kundal. Ivy, question. I know itagtag ta ninyo tamo okay ng a lot of information this afternoon, diba? And the whole day. But then pas-pas trabaho kayo or what? Oo, kasi gipili lang din ako ang mga importante, no? So, by next, ano, and in your post-test, it would be very general. Like for genetics alone, only 5 to 10 questions lang po lang gawas. Diba? Out of 100. Syempre nasa content and outside. For the post test, gikan sa dere, no? But sa ato ang mock board, it will be from all, no? Kasi we will be using, we'll be extracting questions sa ubang reviewers. Sa reviewers from BSU, reviewers from UPLB, and others. So, i-combine lang namo. And then, like as I told you, we will be extending our services until you got the 80% target of passing score sa post. So, as we continue, we will have another step hanggang makuha nyo na tong 80%. Kasi it's the only guarantee na ready na ta. Ready na mo para sa board exam. We still have enough time naman noon. No? After, kasi matapos atong session based on the calendar that we have on the first week of November, 5 or 7. Tapos na ta. So, if, di ba, like sa Ansay, tagilid pa itong score, 50% ratong passing, right? Oh, so, balikan ako. Sa post-test, tanaw na ako. But sa post-test, if 50% gap, at least 50% mo, kasi, daghan man na bagsak sa Ansay, wala ka pa 50%. No, ang kuan ng ana is, if you have much time, practice mo exam na questionnaire. Katong gihatag na mo na questionnaire, na mo'y printed doon, ibalik sa inyo, pwede naman ito i-re-exam. Oo. Re-exam lang to siya. Isulatan na ninyo, di ba sa answer sheet man, isulat ang file. I-re-exam lang to, and then, para mahinumduman ninyo ang answer, and then, ana lang man ang technique, balikon nimo ang exam, bahalag the same set of question, para makuha na to ang 80%. Especially sa soil science, balik-balik rin yun ang exam so far. Nagbalik-balik na. So, I guess yun lang, and also sa mga online, if you have questions, Sir Ron?
None, ma'am.
Ma'am Ernaline and Madam Nova. Do you have questions or clarification? Wala pa ata. Introduction pa lamang ko ang genetics. Introduction pa lang niya siya. So, katong mga types of breeding, cross-breeding, ato pa itong i-discuss. Pero kabalo naman mo, diba? Sa animal breeding? Ha? Animal breeding and plant breeding. Kasi duha lang man na cross breeding. Sa plant propagation dito man nato ang plant breeding. So yun lang. Thank you very much for the huge number of attendance we have today, and hoping tomorrow the same paces o madagdagan pa, no? Katong uban nga nag-online, maka-reach out like kay Ivy. Available ka, Umabing? Dili na. Sige. Ha? So, wala na ba tayo concern? So, ang sir, gihapag na copy sa inyo, ha? So, again, thank you. And kung wala na tayo mga questions or clarification regarding sa ating topic for today. So, katong mga, hormones, ha? Don't forget the 5 major hormones and their uses, as well as mga, simple na lang, gamay naman to actually mga plant adaptation, phototropism, Gnostic tropism, and Gnostic movements. So, maulang ko ni Sir Pundal. Kaya musayaw na si Sir Pundal. Ito na ko, pwede nag-dismiss na. And see you again tomorrow. So sa online naman, thank you very much and have a good day. Indak na punta sa Kadayawan tonight.
Salamat, ma'am.
See you tomorrow. Bye-bye.